Hello all, I'm Alex Allman, and I am a PhD candidate at the Natural Resources Institute, which is part of the University of Greenwich. When preparing for this talk, I was reminded of the prevalence of viruses in our media today with COVID and the recent monkeypox pandemic. Today, I will be talking about the benefits of viruses as part of my project, Viral Diversity and Coevolution with Fungal and Oomycete Pathogens. Fungal plant disease is an ever-present threat to our crops, with diseases such as coffee leaf rust and Panama disease or bananas, threatening some of our most important food crops. This threat is growing due to climate change and the global trade of crops. There is a need to counter this growing threat. To understand what fungal viruses are, we first need to understand the basics of virology. A virus, by its most simple definition, is an infectious agent that can only replicate within a host. Viruses are highly abundant and widespread biological entities with approximately 10 to the 31 individual viruses on Earth. Virology is a quite new science, with the first discovered virus being the tobacco mosaic virus in the late 19th century. So viruses are often portrayed as uniform organisms with little difference between them. However, in reality, viruses are highly diverse organisms, and this diversity is represented in their genomes. Viruses can possess either a RNA or DNA genome, and their genomes are represented by 7 groups. Some of the most important viruses are present within these groups, such as the polio virus represented in group 4 and the influenza virus represented in group 5. So what is a mycovirus? So a mycovirus is a virus which infects fungi. The first discovered mycovirus was the mushroom virus X in button mushrooms and was the cause of Lafrance disease. You can see the effects of Lafrance disease on the lower left. You can see the difference between the healthy, unaffected button mushroom and the diseased, infected button mushroom. So Cryptonetra parasitica virus 1 was the first successful use of a mycovirus to control a fungal plant pathogen, Cryptonetra parasitica, which infects chestnut trees. Only 250 mycoviruses have been discovered so far. Despite the great diversity in despite the great diversity present within viruses, most mycoviruses only possess an RNA genome. Since most mycoviruses possess an RNA genome, it's important to talk about the gene RNA-dependent RNase polymerase, or RDRP. So RDRP proteins are essential to all RNA viruses, as they are essential for their replication. Therefore, the RDRP gene can be used to detect RNA viruses and most mycoviruses. So fungi are formed of multiple cells called hyphae. These hyphae form the basis of our fungal colonies. So mycoviruses are able to spread within these hyphal networks and therefore are not exposed to their outside environment. Therefore, most mycoviruses are transmitted intracellularly. Due to the lack of environmental pressures, mycoviruses are simple even for virus standards, with mycoviruses belonging to the Nanaviridae being the most simplest, only possessing an RDRP gene, lacking the other genes found in other viruses such as the protein coat. Below is an example of one such nanovirus, which is only 2,514 nucleotides long. Unlike viruses of other kingdoms, many microviruses have no effect on their host. You can see on the lower left is an image of Cryptonetra parasitica on a chestnut tree. This Cryptonetra parasitica is not infected by CH3-1, but instead it's infected by the avirulent CH3-4. More interestingly, some microviruses like Saccharomyces cerevisiae, virus LA, can benefit their host by allowing for the production of killer proteins. You can see this in the lower right image, where you can see the infected yeast is not surrounded by any competing yeast. So the reason why this project is interested in microviruses like CH3-1 is due to their negative effect, and this makes them good candidates for biological control, as they are able to reduce the ability of its host to cause disease, thereby disarming it. This ability is called hypovirulence. You can see it in effect in the lower left image. You can see the virus-free colonies in orange, and you can see the contrast that the virus infection has with the colonies in white, representing the virus-infected colonies. So advances in sequencing technology have allowed for this project to occur. So quite notably, sequencing technology has reduced the cost of sequencing from, for example, $10,000 per raw megabase of DNA sequence down to the tiny cost of $0.1 dollars per megabase of DNA sequence. Not only this, the generation of data is much easier compared to conventional sequences. Here on the lower right, we can see that the DNA microarray FEHG has generated a consistent number of samples throughout the years. But after 2011, we can see a growth in the number of samples generated each year with the introduction of the Lumina, HiSeq, and the NextSeq sequences. So this advances in sequencing has provided the data required for virus surveys. Projects similar to mine studying other kingdoms, such as marine life, have discovered thousands of novel viruses. The main advantage of such an approach is that projects such as mine do not require the traditional knowledge of viral biology, such as the host range, in order to find viruses. So just to break down the project objectives. So the first objective is studying what microviruses are present in the genus Fusarium. The second objective is to study where are these microviruses found. And the third objective is to study the barriers to transmission in fungi. So today I will be talking about what microviruses are present within the genus Fusarium. The genus Fusarium contains many significant plant pathogens which are of great threat to food security, such as Fusarium graminearum, which is the causative agent of Fusarium head blight in wheat, as well as other Fusarium pathogens such as Fusarium oxysporum, which is a generalist which can infect a wide range of different crops, including tomatoes and bananas. As of 2019, 29 Fusarium viruses have been discovered and are assigned to 21 different families. So I'm working with RNA sequencing datasets, which contain many small sequences generated by the sequencer called reads. Our pipeline is first required to remove any fungal reads from the analysis, and then the pipeline is required to reconstruct the remaining reads into full-length sequences called contiguous sequences, also known as contigs. These full-length contigs are then used to detect viral sequences by detecting the viral RDRP protein. So 805 RNA-seq datasets were downloaded from NCBI. Any bad bases or erroneous bases were removed from the analysis. and any fungal reads were removed by mapping these all reads to a group of Fusarium genomes. The remaining reads were assembled into contigs, and these contigs were used to predict the presence of the viral RDRP gene to detect any viral sequences. From this pie chart, you can see that the results were highly promising, as within 805 RNA-seq datasets, I detected 434 viral sequences represented on the chart on the left. Within these 434 viral sequences, over 69 virus species were represented, some of which are actually novel. Another exciting result is the high number of viruses from other kingdoms detected, especially the high number of plant viruses, which was 27. This could be down to contamination because a lot of the RNA-seq datasets which I've used were from plant pathology studies, so they would contain plant material. But some could represent a close connection that plant viruses have with mycoviruses, as they have been theorized to share an evolutionary origin together. This will obviously require further research. Certain families were selected for phylogenetic analysis. This was based on whether they contain possibly novel viruses or if they have any connection with hypervirulence. For example, the families Nanaviridae, Alphiflexiviridae, and Petitiviridae, as well as Oomycete. So here is an example of one of the phylogenetic trees which I built. This is for the family Nanaviridae. You can see that the contigs are in red and the hypervirulence, the known hypervirulence inducing mycoviruses are represented in green. Basically, what this phylogenetic tree tells us is that yes, these contigs are novel, which is exciting, but unfortunately, we cannot link any of these novel viruses with the ability to induce hypervirulence yet. So as for my conclusion, microviral diversity is still underestimated. But however, from a genus level study across 805 datasets, we've detected 28 microviruses, 4 of them being novel, just proving the concept that microviruses are rare. Some of these microviruses could even have the potential to act as biocontrol agents of fungal plant disease. I would like to finish with thanking the British Mycological Society for inviting me. I would like to thank my supervisors at the University of Greenwich and the NRI, Dr Andrew Armitage, Professor Cecile, Dr Goncalo Silva. I would like to thank my colleagues at NRIPS, as well as my external collaborators at the University of Kent and NIAB. Thank you for listening.
Fantastic. Thank you, Alex.
Whoops.
Thank you, Alex. Thank you very much. So has anybody got any questions for Alex? If you have, can you please pop them into the chat? If you can't think of any right now, that's absolutely fine. Or if you're still kind of mulling things over, just pop them in the chat anytime you like, because Alex will still be here at the end to cover anything. So there's nothing. There's a round of applause from Scarlett. Thank you, Scarlett. Oh, and Tom as well. So there's a few signs of appreciation from the audience, which is great. Very well-rounded talk. Comment from Cam there. No questions from him.
Yes.
Here comes some questions. So from Victoria, thank you, Victoria. Of the viruses you have found, are any potential biological control for Fusarium species?
Potentially. So unfortunately, since my project is bioinformatics and we don't really understand how the ability of hypervirulence is induced, we can't actually discount viruses having this ability. So until we actually experiment with some of them, we'll figure out.
Okay, so it's kind of the next step in the process there once you've worked.
Yeah. And it's very strange phenomena. So mycoviruses can induce hypervirulence in some strains, but it can do nothing in others. So it is really up to debate. So.
Okay, thanks, Alex. There's more signs of appreciation from the audience. It's fantastic, thank you. So if there's any more questions, as I say, drop them into the chat. Perhaps if we can, oh, beg your pardon, one more question just popped in from Tom. Thank you, Tom. Is there an analogue of viruses causing hypervirulence in other taxa?
In other,
not mycoviruses.
Oh, I see. Like other taxa, like plant viruses. There is a range of different spectrum. There's a spectrum of like different effects that viruses can cause, depending on the kingdom. But hypervirulence is definitely something associated with mycoviruses, as a lot of other viruses would just kill the host, it won't do nothing, or it could benefit. But there isn't anything really comparable to hypervirulence in a lot of viruses.
Thank you. Okay, so we'll move on to Victoria now. Norman, do you want to introduce?
Yeah, this is Victoria Armer from Rothamsted Research, and she's going to discuss unlocking the door how Fusarium graminearum exploits Plasmodesmata during host tissue colonisation. I can't believe I even said that. There you go. Victoria?
Sorry.
What?
Oh, sorry. Very, very quick. All about to start. Okay. So yeah, good evening, everyone, and thank you for the invite this evening. So today, my presentation follows on quite nicely from Alex's actually, because I do study Fusarium graminearum, and this is one of the Fusarium species that infect wheat. And I look at how this pathogen uses the wheat's own archaeology and to basically facilitate its own infection and proliferation. So I will move on. So I first thought I would give a little introduction to myself. So I did my master's at the University of Bristol, and then I went down the road to the University of Exeter. And here I am doing a PhD, although hosted by the University of Exeter, I am based at Rothamsted Research. And this is part of the Southwest BioDTP, which is funded by the BBSLC. And Rothamsted is a great place to do great science. This is one of the long-term experiments we have on site. This is called the Broad Book experiment, and it's been running since the 1850s. And this is looking at different agricultural applicants, such as fertilizers, and the effect it has on wheat yields. We also run the insect survey. So we have traps all over the country and volunteers, and this is a suction trap, which is essentially trapping aphids during the growing season. And they're also transmitters of viruses. So this is all ties into the theme of farming. But we also have amazing lab facilities. So this is where I come in, and I work in this building, the 17-week building. So I thought I'd bring this back a little bit to why fungal pathogens are really important. They're very well studied because the impact of them is so great. And this paper came out about 10 years ago, so back before I even went to university. And this was asked plant pathologists around the world, what do you think are the most important plant pathogens? And they came up with a list. I didn't include the notable mentions, but as you can see here, Fusarium actually features twice, unlike any other pathogen, although not at the top. I would argue it should be at the top. So I study number 4, Fusarium graminearum, but as Alex also introduced, a close relative, Fusarium oxysporum, is also a very serious plant pathogen. So Fusarium causes one of the main causal agents, along with Fusarium colmorum, of Fusarium head blight on wheat. And this is, so all the Fusarium species are ascomycete filamentous fungi. There are very many of them. They're often found in the soil, and they are mostly plant pathogens. So this large species complex infects a huge range of crops and actually is of huge economic importance around the world. So Fusarium oxysporum on tomato, there's vascular wilt here, and also, as Alex said, Fusarium oxysporum also causes Panama disease on banana. You've probably seen it in the news. It's a very serious pathogen. Also in coffee, Fusarium xyloides was another vascular wilt. But we do have one good one. So Fusarium venenatum is the only non-pathogenic species in this complex, and that is the corn fungus. So I promise you it's got no toxins in it. It's very good. So back to Fusarium graminearum. So on the right here, we have a picture of an infected field, and it's demonstrating the characteristic symptoms of Fusarium head blight. So we get this, the blighting, just the bleaching of the wheat head when it's infected. And this is essentially caused by the plant cells becoming devoid of any contents as the fungus infects it. And once the head blight is infected, the infection will move down vascular elements. So these are the phloem and the xylem, so where the sugars and water are transported throughout the plant, basically to infect further. So Fusarium graminearum specifically infects small grain cereals. It's not just wheat, but also oats and barley. And the main impact of it is not just reduction in yield, but also reduction in grain quality. The biggest problem is the contamination with mycotoxins. So Fusarium graminearum is very good at producing this, which is deoxynivalenol. We call it DON. And this inhibits protein translation by binding to ribosomes in eukaryotic cells. So it has a very broad impact, and it means that although even if we can't eat it, livestock can't eat it either, and it survives brewing processes, so we can't even use it for malting. So when we have epidemics of Fusarium graminearum in fields, we can lose 50 to 70% of the wheat harvest. So wheat in grains in storage will essentially allow the Fusarium to proliferate and cause even more damage. So how bad is it? Well, it's got an expanding geographical range, so it's becoming more important. And this is mainly due to the increasing number of warm, wet summers. I know it doesn't feel like it this year, we're in another heat wave in like a month. But we are generating, through climate change, conditions that are more conducive to epidemics. And as you'll see here, we've never settled to pre-epidemic levels of disease. I'm a little bit young to remember 1998, but I remember 2007 and 2012 being very, very wet summers. And as you can see in between, we've never gone back down to the pre-98 levels of disease. However, this is also due to alternative hosts being introduced into the wheat crop rotation cycle, namely maize. But we, monitoring is also increasing, but it's still a huge problem. So, biology. So I have a lot of time to look at the infection biology of Fusarium in wheat spikes. So very lucky here to be able to actually study in wheat. We have all the facilities at Rothamsted. And when a Fusarium spore will land on a wheat spike, so it will do this mainly through splash dispersal, hence why the warm wet summers are very important. And it will germinate on the surface, and then it will have some hyphae that will make its way into the structure itself. So on the far left there, you'll see a spore. They look like little bananas. They actually do look like little bananas. Moving down towards the developing ovule, which is number 3. And then it will move into the rachis node and then throughout the wheat spike. So what it's mainly looking for here is these developing ovules. They've got soft walls, they're not fully formed grain, and they're just full of lots of nutrients. On the top panel here, you can see a kind of cartoon of these monohyphae, and then they'll start to form these infection cushions, and this is where they start to produce a lot of toxins. So as I introduced before, deoxynivalenol, this toxin A, effectors, so little proteins that will cause damage within the cells, and also a lot of cell wall degrading enzymes. Sometimes they will go through natural openings, such as stomata, and then start to grow between cells that call these apoplastic growths. So when they start growing between cells, we call that the symptomless. They are still growing through the tissue, but they have not broken into any cells. And we start to get symptoms when we start breaking into cells. So this is when we start causing a lot of damage, and the cells start devoiding themselves of content. So all the water, the cytoplasm, the sugars in the vacuole, all of that starts to get destroyed by the fungus. So what does this look like on a wheat spike? So on the right, you can see at the top, we've got the symptomatic phase, this start bleaching, so we get this necrosis. And then as you move down the wheat spike, you can see at the very bottom that the Fusarium hasn't quite managed to reach the final H there. So this is the beginning. This will probably be about 5 to 7 days after infection. You can see that we inoculate in the middle. This is black marks on this wheat spikes, will show us when we essentially inoculated with the Fusarium spores. So I introduced at the beginning Plasmodesmata, and Fusarium exploits Plasmodesmata when it's after it's broken into its first cell, it will start to grow between cells using these structures. And they're very simple. They're composed of an endoplasmic reticulum, and these are normally found in other areas of the cell. This is a very small one, and it also has some structural proteins that aren't pictured in here, but they control substances flowing between cells. These what connect cells together. So in a leaf, you will have lots of cells that are connected through these. And this will allow the transport of ions and sugars, sometimes very small proteins, sometimes some salts. But they're very important for both plant development and also plant signaling. However, these are weak points within the wall. So these cross the cell wall, and they also they're weak points within the cell. So what do they look like? Well, they're so small we can't image them using light. In fact, they are smaller than the wavelengths of light. But on the left, right here, we have at the top an electron micrograph from a group at the John Innes Centre showing Plasmodesmata in a what's called a pit field. So they'll often congregate together in the cell wall and usually found in pairs. And on the right, bottom right, there is a picture of some Plasmodesmata which have been tagged with a green fluorescent protein. And we can see this one. This is big enough for us to see. And it shows just how numerous these are within a leaf cell. So fungi aren't the only pathogens that use Plasmodesmata, and they're a general weak point for in plant cells. As you can imagine, breaking through a cell wall takes a lot of effort. So once you've got in once, you want to just keep using these weak points. So viruses use them quite often. So fungi use them, and also bacteria. So this is well reported on. But plants have developed something towards their defense mechanism, and this is the use of callose. So callose is a substance that can be adjust the permeability of these channels, and they can essentially block them off completely and prevent any of the effectors or toxins from the invading pathogens entering into healthy cells. So we call this symplastic isolation. And this is very important for what I'm looking at. So this is what Fusarium crossing one of these Plasmodesmata looks like. As you'll see in the middle panel on B, the invading hyphae constricts quite dramatically before crossing the cell wall, and then will take back its original shape. And you see in panel C, these Plasmodesmata do actually come together, as you saw in the electron micrograph. And so these hyphae on the right are quite old, so they're starting to get large vacuoles and lipid droplets within them. But essentially, this is an essential part of Fusarium infection biology. So I'm starting to question, can we increase host defenses to stop this happening, either to slow down infection or prevent it completely? And a way I'm doing that is using this technique. This is also seen in this paper, but essentially I am inoculating wheat spikelets, and I use a lot of microscopy to have a look at what's going on inside the tissues. So I freeze everything in the cell using paraformaldehyde, and then I fix these into resin. That involves a step of ethanol dehydration to get all the water, and then we infiltrate with this resin. We bake it overnight, and then we take very, very thin slices, they're called ultra thin sections, and they're a micron thick, with a glass knife, and then we stain them with toluidine blue. And this allows us to see cellular components, but mainly what I'm looking at is the cell walls. As you might notice within this, all of these cells are empty. It isn't just they aren't being stained. You'll see fungal hyphae, which have been frozen in place, and the cell walls, but you can't see any cytoplasm or other organelles. You might also see nuclei in there, as well as chloroplasts. So these are now gone. So this is far through the infection. And as you can see, it's very, very infected with fungal hyphae. They are in the purple, and then the cell walls are in the blue. And depicted with these yellow arrows here are Plasmodesmata crossings. They're very numerous, and they are clearly a very important part of this infection. So the crossing of Plasmodesmata, as you might have gathered, only occurs during the symptomatic phase of Fusarium infection, but it's still a very essential part of this infection biology. It also correlates with extensive release of cell wall degrading enzymes and toxins. So clearly, finding a Plasmodesmata is not enough. You need to attack it. So as you see, this intermediate phase is where we start to see the Plasmodesmata crossings, but during the symptomless phase, we wouldn't have any. So how do I study the role of Plasmodesmata? Well, I mostly work with Fusarium. So I would start testing a range of single gene knockouts. So we'll knock out parts of the infection process that we think are important. And we try to make it really hard for Fusarium to cross these Plasmodesmata. And the idea is that we can start to find components which are being attacked and then try to upregulate them in wheat. So can we then start to identify genes in wheat that we can breed into resistant varieties to kind of make it very hard for Fusarium to infect these, and most importantly, produce loads of toxins which you really do not want in our food. So yeah, thank you very much for having me. That was a very whistle-stop tour, I suppose. So obviously my lab in Rothamsted, my supervisors, of course, and funding, and with the British Mycological Society for having me here. Okay, I'm sorry. And yeah, any questions?
Thank you very much, Victoria.
That was very fast.
That's great. So yeah, so good comments again in the chat. Thank you very much, and saying how it paired very well with Alex's talk, which it certainly did. So if anybody's got any questions, if you can pop them in the chat in the next minute or two, that would be great. While you're doing that, I have to send apologies from our third speaker. I'm afraid whilst we've been listening to the first two talks, Nilu has been in touch to say she's unexpectedly unable to join us this evening. She sends huge apologies, especially if you were waiting to hear her talk specifically. We do hope to rearrange that talk for another time, and I'll be getting in touch with Nilu another time to do that. So again, please accept huge apologies from Nilu that she can't make it this evening. So now I'm going to go back and have a look at the questions. Okay, thank you. So first of all, from Scarlett, how much of a threat is Fusarium to food security? If we don't find a way to control Fusarium, do we need to be worried about wheat crops in the future?
So it is a very big problem. It's actually not so much of a problem in the UK. It's much bigger problem in the US. As I mentioned very briefly there about introducing corn into cropping cycles, the US obviously subsidized corn, so that is a huge part of farmers' income. I think it's hard to predict what's going to happen in the next 10 years. If we start having more dry summers, then we won't be fine. And we are producing more resistant varieties of wheat. One of the problems with Fusarium is that it's resistant to almost every single fungicide under the sun, so we cannot control it with fungicides. Yes, that's why we're studying weeds. It is a problem, but there is so much research in this area that I'm sure we'll be able to find a solution. We have a lot of genetic diversity in wheats, whereas with banana breeding, it is a lot bigger problem because you might know that all banana plants are clones of each other. So there is not a lot of genetic diversity there. So, yeah.
Okay, thank you. Tom says, do you have a handy list of which genes are knocked out in your experimental fungi? I don't know whether Tom means handy for him or handy for you, but is it handy?
No. So it depends what you're looking at. I think I might be one of the only people looking at solid-graining enzymes. It's quite hard to isolate one that is the only one. Often when you take out one thing, something else is going to be upregulated and take its place. So yeah, maybe by the end of my PhD, you can have a look in the thesis and it might be there.
Thanks.
But not at the moment, no.
Okay. From Danny. Hi, Victoria. Have you had any luck with managing to upregulate the defense mechanisms, or is this still theory at the moment? And link as second part of the question, linking to Alex's lecture, has the use of mycoviruses or similar been considered?
So the first part is working in wheat is very hard. As you know, it's got a hexaploid genome. So if you want to upregulate one thing, you will have to upregulate all 6 genes, and often there are duplicated genes in there as well. So for me to do that, it would probably be another 2 PhDs worth for wheat breeding. But for me, it's just identifying the targets at the moment. But no, I have not considered the use of mycoviruses at all. It's something I found very interesting, and I certainly haven't heard of it being used for Fusarium species at all. So I'd be very interested to see if Alex found some candidates that maybe we could test.